Hey guys, so today we'll discuss biology paper 1 and multiple choice question, which is May June 2022. Question number 1. The photomicrograph shows a bronchiole and alveoli. The magnification of the image is 360. What is the maximum diameter of the bronchiole lumen? The first for the bronchiole diameter, we need to measure it with a ruler. But here I'm using iPad, so obviously my reading won't be accurate. But for the lumen, you need to take from here till here. So you need to measure this lumen by using your scale, which is basically in centimeter. So you have to convert into meters. This is 5 centimeter. Okay, so 5 centimeter you need to convert it into meter. So it will be 5 into 10 to the power minus 2. Okay, by using the formula magnification is equals to image size upon actual size. We are using this formula because the magnification is given, and the image size we found it by using the scale, so we need to find the actual diameter. So this is 360 the magnification, image size is 5 into 10 to the power minus 2. I convert it into meters and the actual size. So to find the actual size, 5 into 10 to the power minus 2 upon 360, so it will be 1.39 into 10 to the power minus 4. This is in meter, and here the options are given in micrometer. So I need to convert this actual size into micrometer by multiplying it with 10 to the power minus 6. 10 to the power plus 6 is equals to 139. So it will be equal to 140. So the answer is C. Okay, now the next question. Number 2. A specimen is observed twice with a microscope. Firstly using green light with a wavelength of 510 nanometer, and then using red light with a wavelength of 650 nanometer. What happens to the magnification and resolution when using red light compared to green light? So basically, we need to find the magnification and resolution of red light. Okay, so of red light compared to green light. So if the wavelength is more, the resolution will be less. So basically they both are inversely proportional. So here the wavelength of red light is greater than the green light, so the resolution of the red light will be less than the green light. Okay, so this can be C. Why C? Because the resolution decreases as the wavelength increases. Okay, and the magnification remains same because there's no change in objective length, or there's no change in eyepiece. Okay, so now question number 3. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. Produces the mitotic spindle during cell division, synthesis of polypeptides, synthesis of lipids. Produces the mitotic spindle during cell division is basically centriole. Is basically centriole, so it is centriole. Synthesis of polypeptide, so basically rough ER is responsible for this function, so it is rough ER or ribosomes. And synthesis of lipids is smooth ER, or smooth endoplasmic reticulum. V is membrane which surround an enclosed inner cavity. Non-membrane bound spherical structure. A double membrane interspersed with pores, non-membrane bound cylindrical structures, membrane bound sacs arranged as a flattened stack. Which student correctly match the numbered functions with the appearance of the cell structures? So basically, centriole is a non-membrane bound structure, and it is a cylindrical structure, right? Because centrioles are basically like this, right? So it is a cylindrical structure. So why would be centriole? So here we have one which is centriole, so it can either be C or D. Let's see the option number C, whether it is correct or not. W is this non-membrane bound spherical structure. Ribosomes or rough ER are non-membrane bound. It is correct, and it is spherical structure. And it is smooth ER, so it is basically surround and enclosed inner cavity. So the correct option is C. Okay. What is found in chloroplast and mitochondria? Chloroplast is basically found in prokaryotes, so it will have a ribosome which is 70S, and it will have circular DNA. Also, mitochondria also have circular DNA and 70S ribosome, so it will be B. Which feature is correct for all known viruses? So basically, caspase made up of lipid and protein. It is wrong because caspase is made up of protein, not lipids. DNA core, it may contain DNA or RNA, so it is not the correct option. Outer envelope of phospholipid, no. So the answer is D. The correct answer is D, non-cellular structure. 4 extracts from different plant materials were made and tested with Benedict solution. The extracts were boiled with Benedict solution for 240 seconds, and the final color was recorded. 1 is red, 2 is yellow, green, 3 is blue, 4 is green. Which sequence of plant extracts represents an increasing quantity of reducing sugar? The Benedict solution is basically blue in color, and if the reduced sugar is present, then it will convert to brick red, or it converts from blue to green to yellow and then to red or brick red. Okay, so the sequence represent an increasing quantity. So the increasing is the most reducing sugar color is red, so it will be 1, and so the answer is D. Which have properties that are dependent on hydrogen bonds. Number 1 is cellulose, a molecule of hemoglobin and water. All of them are dependent on hydrogen bonds, and the answer is A. Which statement is correct? Cellulose, glycogen, and amylopectin are all polymers. It is correct. Ribose, amylase, and phospholipid are all macromolecules. Ribose is a monosaccharide, not a macromolecule. Starch, glucose, amylose are all monomers. Starch is a polysaccharide. Sucrose, deoxyribose, amylopectin are all polysaccharide. Deoxyribose is again monosaccharide. So the answer is A. The diagram shows 2 amino acids. Some of the hydrogen atoms are numbered 1 to 6. Which 2 numbered hydrogen atoms could contribute to the production of molecules of water when a peptide bond forms between these 2 amino acids? So a peptide bond is basically formed between a carboxyl group, which is this one, and this one, and an amine group. So this is the amine group, and this is the amine group. So a peptide bond, or the 2 number hydrogen atom that will contribute to the formation of water in the peptide bond. A peptide bond is formed either between 3 or 4 or 6 and 1. So it will be B. A student wrote 4 statements about water. Water has high specific heat capacity which maintains the temperature of water within cells. It is correct. Mammals rely on water having relatively low latent heat of vaporization to keep them cool. Mammals have high latent heat of vaporization, not low. When a negatively charged ion is added to water, the delta positive charge on the hydrogen atom is attracted to the ion. It is correct. And when surrounded by water, the nonpolar molecules tend to be pushed apart from one another. When surrounded by water, like for example in a phospholipid bilayer, when it is kept in a water, it forms a blob or it forms a round ball with hydrophobic ends facing each other. So basically the hydrophobic ends or the non-polar molecules are pushed pushed towards each other, not push towards each other, not away from each other. So it will be um B, because 1 is correct, 2 is not correct because it requires high latent heat of vaporization, 3 is correct, and 4 is wrong. Typical enzymes are large globular proteins with a specific tertiary shape, which molecular interactions are directly involved in maintaining the tertiary shape. So basically for the tertiary shape, all these bondings are required, and there's one other bonding, that is ionic interactions too. Interactions. So it will be A. Which statement about the Michaelis-Menten constant is correct for an enzyme with a low affinity for its substrate? So basically the Km is inversely proportional to the enzyme with a low affinity. Okay, so here the enzyme has low affinity. Enzyme having low affinity means that that substrate binds to the enzyme not readily. So the it has a low affinity, so the Km will be high, so it can either be A or B. Now let's see the Vmax. The Vmax should be high at the Vmax should be at a high substrate concentration, because the substrate are not readily bind to the enzyme, so they need a high substrate concentration so that they can bind to the enzymes more readily to reach their Vmax. So the answer should be A. Long chain saturated fatty acid change from solid to liquid at higher temperature compared with short chain unsaturated fatty acids. Which fatty acid would be more likely to form triglycerides in mammals that live in cold climate? So basically, in cold climate, the fluidity decreases. Okay, so basically to increase the fluidity, or when fluidity is increased, when there is increased saturated carbon, there's increased saturated carbon, and there's increased short chain. These two conditions will increase fluidity. Okay, so in cold climate the fluidity is decreased. So to compensate this, you need to have short chain which are unsaturated. Okay, when animal cells are cultured, salt solution is added to keep the cells alive. What is the purpose of the salt solution? to allow facilitate diffusion of salts into the cell, to prevent diffusion of other ions in or out of the cells, to prevent net movement of water in or out of the cell, to provide a source of energy for active transport. It should be C, because you need to keep the cell alive. So to keep the cell alive, you need to prevent the cell from cell lysis or cell bursting, or from cell shrinking. So we can and when the cell burst, when there's increased water coming into the cell, or when does the cell shrink, when more of water goes out of the cell. So to have a normal, or to have the net movement of water in and out of the cell, will prevent the cell from all these bursting, lysis and shrinking, so the cell will be easily kept alive. So the answer will be C. The following are all processes that allow movement into cells. Which processes require ATP? Phagocytosis require ATP. Active transport require ATP. Cellular diffusion does not require ATP, but it does require carrier proteins for transport. So it will be A. Which features are required to allow for efficient diffusion? A large surface area, yes. Surface area is required for diffusion. A short diffusion pathway, yes, because the shorter the diffusion, the more it will diffuse readily. Like for example, in alveoli, the layers or the membrane the oxygen passes through is less. That is why it diffuse easily and more rapidly, uh, and maintain a constant diffusion gradient. Gradient is required to be maintained, because if there is no diffusion gradient, then the cell or then the oxygen won't diffuse into the correct direction, because diffusion occurs from higher concentration gradient to lower concentration gradient. So it will be A. What is the role of mitosis? The role of the growth of organism, yes. Production of genetically different cells, no, because mitosis is genetically identical cells. Repair of cells, yes. Replacement of cancerous cell, no. The growth of organisms, yes, because during cell division there's asexual reproduction, where the cells are divided identically. Like for example, during fertilization zygote form, and when zygote divides, it divides by mitosis. And C is also correct, because repair of cells, because once the cell die and the new cells are being repaired, they are repaired identically. But here we need to answer the best option, so it will be A. Question number 18. Prevent the loss of genes from the ends of chromosome during DNA replication, but they become shorter each time they are copied. In cancer cells and stem cells, the telomeres remain the same length. Which statement is correct for all human cells? If telomeres become short, a cell may stop dividing. Yes, because telomeres are basically end DNA sequence. If And by each replication, they become shorter and shorter. So if they become shorter and shorter, the genes won't be copied, and the cell eventually stop dividing. So A is correct. Now let's see B. Adding telomeres could increase the rate of aging. It would decrease the rate. Okay, it will decrease the rate. Telomeres are repaired by the enzyme RNA polymerase. It is repaired by telomerase. Telomerase. Telomeres prevent all damage occurring to DNA. Not all. Okay, so the answer is A. Question number 19. The nucleus of a mouse body cell in G1 phase of the cell cycle has 1.2 into 10 to the power minus 12 gram of DNA. What will be the mass of DNA in the nucleus of the cell at the end of S phase and at the end of G2 phase of the cell cycle? So basically in G1 phase, the cell grows in size and synthesize messenger RNA and protein that are required for DNA synthesis. The cell did not start DNA synthesis, and in S phase the DNA synthesis occurs, so the gram of the DNA would be double because exact copy of the DNA is being formed, so it would be either C or D. And at the end of the G2 phase of the cell cycle, so basically in G2 phase cell prepare itself for mitosis, so there is no replication, no synthesis, so the weight or the number of DNA would be same, so it will be D. What occurs during prophase in animal cell? During prophase, it is basically all the structures are being disappeared or destroyed. So first, number one, fragmentation of the nuclear envelope. Yes, because the nuclear envelope will break down into fragment and then eventually it will disappear. The nucleus disappears. Yes. Stained chromosome become visible. Yes, because they need to undergo into metaphase, anaphase, and then all those stages and then divide. Centrioles replicate. No, centrioles do not replicate, so it will so the only correct options are 1, 2 and 3, which is B.